So let me start by putting this in the context of how this study fits into a much broader initiative. So the initiative is called the Collaborative Initiative on Fetal Alcohol Spectrum Disorders, so on FASD, and the consortium is called FASD. I've made a point of showing you the website on the very top, but also importantly, there's a schematic or a visual of all of the investigators that are involved in this study. And as you can see, there's a number of individuals. We look like we're in a lovely location. And so we meet frequently, but importantly, as you look across that whole set of faces, what you see are individuals looking at the question of FASD and FASD research from a number of different areas. We have people who are looking at very basic animal models, trying to understand through an animal model how alcohol in a prenatal setting affects outcomes. We have individuals who are looking at imaging data. We have investigators that are looking at this clinically. And we also, very excitedly, in the latest round of this study, also have investigators beginning to look at interventions. And so it's a really broad team, a great collaborative group, really in many different ways trying to tackle the questions inherent in fetal alcohol syndrome. So let me start by stating a little bit of obvious things so that we're all in the same place. So fetal alcohol spectrum disorders, or FASDs, those are a group of conditions that can occur in an individual whose mother drank alcohol during pregnancy. The effects can be very varied and can include physical problems, behavioral and learning problems, and often an individual with an FASD can have a mixture of these findings. And importantly, we'll also talk about individuals where it's not obvious that they're having any difficulties, or perhaps some of the problems are more subtle. And that really leads us to what many people think of first when they think of a fetal alcohol spectrum disorder, which are some facial features. There are some characteristic features that are often observed, and this is how sometimes a diagnosis is made. So there's knowledge that there's some prenatal alcohol exposure, or there might have been, and then an individual might have some characteristic features. So those might include a smaller head. You can see a couple different changes that occur around the eyes. So some small eye openings, or what are called epicanthal folds, right around the inside rim of the eyes. A short nose, what's called a flat midface, so around the cheekbone area, a thin upper lip. You can see the smooth philtrum, and you can also see what's described as an underdeveloped jaw, or sort of a smaller chin. Those are things that when people talk about fetal alcohol spectrum disorders, often if someone's seen clinically, these are the features that someone might be looking at. But when we think of an FASD and we think of fetal alcohol spectrum disorders, it's really an umbrella term. It encompasses a much broader range of what we would call features. And so if you look here on the left, we've got our umbrella right there, and we start with blue, which is fetal alcohol syndrome, which is sort of the most visible form of the disorder, something that is the most commonly thought of. So those are individuals that have the facial features I just described, often are smaller for size, and also have what we call central nervous system abnormalities. So those might be some of the behavior findings, some brain changes, a characteristic set of features. But these are often kind of the most extreme findings that we see. There are a number of individuals who might also have what we call partial fetal alcohol syndrome, where they meet some of these features, but not all. Then we go to sort of that green part of the umbrella. These are people who meet what's called alcohol-related neurodevelopmental disorder. These would be individuals that perhaps don't have the facial features we so typically think of when we think of fetal alcohol spectrum disorders, but perhaps have some changes that we can see on MRI in the brain, or perhaps some central nervous system abnormalities. So we have to look a little bit more detailed in order to be able to document some problems. And then we have what's called neurobehavioral disorder associated with prenatal alcohol exposure, or NDPAE. And those are primarily individuals that have behavioral effects of prenatal alcohol exposure. And the reason I walked you through that is I want us all to sort of think about how much variability there can be among individuals who have fetal alcohol spectrum disorder. What's in common is some prenatal alcohol exposure. But what we see when we look at that individual, importantly, what a clinician or a healthcare professional might see, and when they're trying to understand what kind of difficulties the person has, it can be very, very different what they're looking at. And what we'd like to understand is what contributes to this variability? Why are there so many different outcomes among individuals who might have been exposed to alcohol prenatally? And the reason we care about that is we think if we understand FASD better, that could perhaps give us some important information that we can use to develop improved interventions or improved treatments. And that ultimately is the reason we do this research. We really want to be able to help outcomes for individuals who've been exposed prenatally to alcohol. So we start to ask, what could be some of those factors? What might be contributing to the variation we see? Individuals who've had similar patterns of prenatal alcohol exposure, why are there different outcomes? Well, there are a couple things that right away come to mind. The first thing we think about is the environment in and around the mother. And you might say, what do I mean by that? Well, this could be the environment, something like nutrition for the mother. It could be things like the health care that she has. It could be a number of other factors in terms of the support around that mother. Genetics might also play a factor, and that's the part that we're exploring in this study. Now, I'll tell you a little bit more about genetics in a moment, but we have to think about the genetics of the mother and also the genetics of the fetus or the baby. So we actually have two kind of pieces of genetics that we need to think about. And often it could be a combination. As with so many things, there's often not just one explanation. Sometimes it's pieces of both that might be important. So let me start with kind of a basic genetics review, just so we're all on the same page as we start to talk about genetics terms. So when we're talking about genetics, we're thinking about DNA, or deoxyribonucleic acid. That's what's often called the genetic code. And we hear a lot about DNA if you read the media, all kinds of things now about DNA. So what is it? It's a long sequence of bases or letters that are arranged like a staircase or like a helix. And we're going to have a picture of that in a couple slides. DNA is important because it codes for something called genes. So genes, if you skip to the last bullet, are things that determine your hair color, your eye color. Genes are important in determining your height. Much of what you are and what you look like is contributed to us through genes. And if you imagine what genes look like, many of us try to imagine them almost like beads on a string, where each one of those beads is a different gene. And to give you perspective, there are about 20,000 genes that all of us have in our body that are encoded by our DNA. So this slide now gets us away from words, and we can think about like a picture. What does this really look like? So this circle represents a cell. We have cells throughout our body. All of us are made up of a number of different cells. And I've shown you sort of these unusual shapes around the edges. Those are all things that are sort of like the workhorses, the power horses of the cell. But I'm a geneticist, and what I care about is the piece in the center in that blue circle, which is what's called the nucleus. And what you're looking at in that picture right there, these are actually chromosomes. These are the chromosomes of an individual, and they look sort of like little different pieces of string. But what we do in genetics is we organize those little pieces of string, those chromosomes, into something called a karyotype. And now we see them kind of organized, and let me explain to you what you're looking at. So everybody has 23 pairs of chromosomes. So if you look at them, they're numbered from 1 to 22. And then at the end, you see those X's and Y's. Now, importantly, we all say you're partly your mother, you're partly your father. And the way that works when we look at chromosomes is every one of those chromosome pairs, whether it's that pair numbered 1 or the pair numbered 2, you get one of the members of those pairs from your mother, and you get one from your father. So when we say that you're kind of half your mother and half your father, it's actually really systematic. It really is half of each. And then when you go look at the end of those X's and Y's, that's sort of the interesting part. The karyotype you're looking at here is actually of a woman because there are 2 X's. So this person's gotten an X from their mother and an X from their father. If this were a man, there would be an X and there would be a Y. We would have one of those chromosomes, one each in each of those categories, and that would make an individual who's a man. So part of the reason I show you these chromosomes are, in some disorders, we can look at chromosomes and we know what the condition is that the person might have. There's a well-known condition called Down syndrome, which means an individual has 3 chromosome 21. So if you looked at this karyotype, there would actually be 47 chromosomes, and there would be a 3rd chromosome in that category above that number 21. But when I look at these chromosomes, I can't tell whether or not this individual will have developed fetal alcohol syndrome or fetal alcohol spectrum disorders. I can't tell whether or not this individual has been exposed to alcohol prenatally. I can't tell any of those things from looking at these chromosomes. And that's one of the challenges that we have when we try to identify whether or not an individual might have been exposed to alcohol prenatally when we don't have a lot of information about that period, or when we're trying to figure out if that person might have been exposed and we're trying to figure out, are the problems that that person might be having, could this be due to prenatal alcohol exposure? We can't tell by just looking at these chromosomes. And there are some conditions where you could figure that out, but this is not one of them. But I wanted to show you these chromosomes and this DNA to tell you that the work we do, though, looks at these chromosomes, but we look at them in the tiny, minute level. What you see across all these chromosomes are actually 3 billion points, or 3 billion nucleotides. And that's how much coverage we have to do when we think about genetics. We have to try to figure out what might be going on across 3 billion different points of the DNA. So how could genetics be important in fetal alcohol syndrome? Well, the first thing is everyone's DNA is slightly different. The only time I would I'd be careful about saying that is if you had identical twins. But if you take two people in the room or somebody else sitting in the room with you, chances are, unless you're identical twins, your DNA is different. And if you're related to each other, maybe a little less differently than someone who's not related to you. But we know or believe that there might be some DNA changes that make some individuals more susceptible or have more effect if they've been exposed to alcohol in utero. Okay? And what we're trying to understand in this study is, where in the DNA might that be important? Where among those 3 billion base pairs might there be something that's affected by prenatal alcohol exposure? So that's the kind of the goal of our study, is evaluating the role of genetics in the variability that we see in fetal alcohol spectrum disorders. And remember, that variability is whether or not the individual has facial features consistent and makes it more easy for someone to realize there might have been prenatal alcohol exposure. Maybe it's an individual who's just having some difficulties with behavior or learning. How can we identify these individuals better, and what might be the role of genetics? So in order for us to try to answer this question and ask this question, we really need to be able to compare large numbers of individuals. The kind of patterns we're looking for, we can't just study 1 or 2 people and understand the role of genetics. Genetics, unfortunately, requires us to compare lots of different people. So our goal is to enroll more than 1,000 individuals in our study, which means we have to have a way to reach out to people and ask them to participate. And that's part of the reason why doing this webinar was really, really a great opportunity for us to let you know about this study that we're doing. So the study I'm going to tell you about, we call it DIG, FASD, and DIG stands for Dissecting the Genetic Contributions, DIG, to Fetal Alcohol Spectrum Disorders. So we really are trying to dissect what's the importance of genetics among individuals who've been exposed to alcohol prenatally. And why we're excited about this study is it's a new kind of study. You don't need to come into a study site or go to another place. You don't need to be evaluated by somebody in person. This doesn't involve having an MRI or doing a number of tests at this time that you'd sit and do on pencil and paper. This is a study that you're going to do entirely from your home. So if you want to learn a little bit more about this, maybe not while we're doing this webinar, but a little later, we have a website, and the website's shown right there. And I've just given you a little snapshot of our website so you can get a feel for what's there. First, we give you a welcome to the study on the far left. Then we let you learn a little bit more about our particular study, but we're also trying to provide links and resources about what is FASD, what's available. And of course, one of the resources we list is NOFAS. We've got more information about research studies people can be involved in. And of course, we want to make sure that you can contact us if you have any questions. So you might wonder, well, who can do this study? Who could participate in a study like this? Well, anybody who's 7 years of age or older. And now if you're wondering why not somebody younger, well, we're going to be talking a little bit about the idea of informed consent. That means you learn about a study and you're able to make a decision about whether or not you want to participate. And the level at which we've prepared our materials is really only appropriate for people down to the age of 7. So a child who's younger than 7, we would need to be able to tell them about the study in a way that we're not quite ready to do yet. So if somebody who's going to participate has to be 7 years of age or older. Because this is a study of fetal alcohol spectrum disorders, we need to either know or suspect that there's been prenatal alcohol exposure. We're going to do this study entirely through the internet, so there has to be internet access. You're going to be coming on our website and doing everything on a computer or on a phone, and so you need to have internet access. I'll tell you about a little later part of the study, but you're going to need to have a way to take a picture. So that means it could be a camera or a smartphone or a tablet, could even be a regular phone as long as you had a way to get a digital camera. But there's options. We can work with you, but you'll need to be able to take a picture. The other thing we're going to be doing is collecting saliva, and I'm going to tell you about that a little bit more later, but we can actually use saliva to get DNA. So that same DNA I showed you, we can make and study our chromosomes. We can do that from a saliva sample. So there's no need to do needles or go somewhere to have blood drawn. And then the other piece is everyone participating in the study needs to be able to speak English. And that's because we here at the study, we only speak English, and so we can't answer questions, and our materials aren't in any other language other than English. So I mentioned this word online consent and an informed consent. So let me explain to you what that means. So we do this again all on a computer, so you need to be able to have access to the internet. And so if the individual is an adult, someone over the age of 18, or someone between even the ages of 14 and 17, you're going to use what's called our online consent. So what I've shown you right here on the left side of the slide is kind of what it looks like. So it's broken up into pieces. It starts by about this research, and we tell you a little bit about it. We explain why we're doing it, and you can read this on our website when you're going through what's called an online consent. Importantly, there are places where you can ask us questions. So you can see on the top, it says, do you have any questions about the information you've read so far? And if you say yes, what comes up then is a box that you can see right below it where it says, type in your question. And we've gotten a question right here that we often get, which is, can my adopted child participate? And the answer is, if it's your adopted child, yes, they can participate. So you, as their adoptive parent, you'll be signing as the adult. The child would have to be over the age of 7 in order to be able to participate in the study. And we realize that that's going to lead you to tell us, have questions. You may not have as much information about prenatal alcohol exposure. And so we're glad to talk through about what information is available, what previous assessments have been done. And we're going to talk about that as we start to walk through the webinar. But I just want to encourage, we want to be able to get your questions, and then we call you and answer your questions. I mentioned that children between the ages of 7 and 13 can also participate. So what we've done there is, rather than having a lot of information that you need to read, we've made a video, and the video has some animation. It has a person who talks and explains things to you, and we use that again for individuals who are a little bit younger, and that's a great way for them to learn about the study. Now, importantly, when we have a child between the ages of 7 and 13, we still need to know that a parent or legal guardian wants the child to participate in this study. So the parent or legal guardian still has to do that online consent, the one that I just showed you that you would read. So actually 2 people need to do that work then. Okay, one doing it on the video, one doing it through that a parent or a legal guardian online consent. So what we do to make sure that you're kind of engaged and the child is listening is there's also some questions. So there's a series of videos. They're all relatively short. And after each one that walks through and tells the child about the study, there's a question. And we've just given you an example here. And you may not know the answer to this question because I haven't told you about this. So I'll just read it now, and maybe we'll talk about it a little bit later again. So the question is, what will you put into the tube that you will get in the mail? You don't know the answer to that yet, but there's 4 choices here. The person who had watched the online assent and watched that video would know the answer. You're going to learn the answer a little bit later in this webinar. So once you've completed the consenting process, the child, either by watching the video or somebody over the age of 13 by doing that reading consent and doing that online. And again, if it was somebody between the ages of 13 and 17, and a parent would or a legal guardian would need to do that too. Then we have some online forms that we also ask you to complete. And I'm going to tell you a little bit more about those in a moment. But these can be done entirely online. Again, you don't need to go in anywhere. You can do these all at once, or you can decide that you want to do some of them now. You can come back and do the rest of them. And if you forget to come back, we'll also send you an email and remind you you've still got a little bit of forms that you want to do. Why do we have these forms? Well, we need to gather a little bit of basic information. The first is what we call demographics. How old is this person? The person who's the participant, the person who we believe was exposed to alcohol prenatally. Are they a man or a woman? what's their race. And the reason we're interested in race is partly this is a study that's funded by the National Institutes on Health, and they want to make sure that our study is accessible to people of what we call all races and ethnicities. We want to make sure that this is really a representative study and that many different people are able to participate. We're also going to collect a health history. So what we're very interested in is, have there been prior evaluations for a fetal alcohol spectrum disorder? Has this child or individual been seen by a physician? Have they maybe been seen by a neuropsychologist? What other evaluations have been done? That helps us kind of put the pieces together to figure out what kind of difficulties or what kind of features does this individual have of an FASD. And then, not surprisingly, we have to gather some pregnancy history information. So it's important if we're able to document a prenatal alcohol exposure, but we also want to know what other exposures might there have been that also might be contributing to challenges that the person might have had. And so we'll ask a little bit about drug and about other exposures as well. And we realize sometimes you may not have all this information, and we just ask you to do your best. Fill in the information you know, and when you don't know, just tell us, I don't know the answer to that information. And the key thing is, we have a fantastic team here. I'm located at Indiana University, and so if you have questions while you're going along or you finish the forms, we're going to give you a call and we're going to want to ask, do you have questions? We want to make sure you're comfortable, know what's going on before you keep going on in the study. And so you get a phone call from us and we'll kind of talk to you one more time and explain the study again. Just make sure we all know what's going to happen next. So what is going to happen next? Well, next we're going to take some photos. So I mentioned early on that one of the things you needed to have was digital camera or a phone or a tablet that you could take photos of. You might be wondering, well, why would I need to take a photo? Well, if you remember early on, I described to you what we often say are some facial features that individuals who've been exposed to prenatal alcohol might have. Not all individuals do, but one of the things we do as part of the study is we take a look at the face of the individual and see whether or not those features that are sometimes seen are present in the individual who's participating in the study. So we need photos to help us understand that, be able to help us try to understand in terms of when we start to look at the DNA, what other facial features might there be or not be that we can start to analyze and look at in the context of that DNA. So one of the challenges if we say, go take a picture of yourself is, who knows, I hate to say it, we're not all equally good photographers. So what we need to do is sort of level our playing field and make sure that the photos we get from different people using different technologies, doing people of different ages, maybe not lining up everybody in exactly the same way, that we're going to be able to compare and use those pictures. So you're looking at these photos right here, or this image of these three people, of this one person kind of from three different angles, and let me explain what you're looking at. Your first picture is the person in portrait, kind of looking at them straight on. Then you're looking at a person with what we call a three-quarter pose. So you see one side of their face a little bit better, but we can still see kind of the front as well. And then you have sort of a classic profile that lets us kind of look at, remember that mid-face to see whether there's a little bit of flattening, gives us a good look at that chin. It's a really great way in each of those photos for us to look at different parts of the face. Now, as you're looking at this picture, you might be wondering, what are those white rectangles on there? Well, those are actually stickers or labels that we send you. And you might wonder, why would I have to have that? Why would you give me those, and why would I put those on my face? Well, if you think about it, when someone's taking a photo, people of different ages, we have to kind of be able to adjust and consider, how big is that person's head? So for those of you who've taken a picture before, if someone comes really close to your camera, their face is going to look really big. If someone stands a little further back, their face is going to look a little smaller. Well, the way we're able to figure out, even though people set up their pictures a little bit differently, how big someone is, how small someone is, those little white rectangles are the same size for everybody, and it helps us get kind of a feel for the size of the person's face. It's as simple as that. The other thing is we then have to be able to get those pictures back from you. So we have a way for you to upload those photos also on your computer, and we do it through what's called a secure website. So that way it's secure. Other individuals can't see that image being uploaded. So again, it's a little bit about privacy of a participant. Now, the next piece that you'll be asked to do is you'll also be asked for a saliva sample. So I mentioned that, and remember that question that you saw earlier, how will you send your saliva sample? Well, now you're starting to see the answer. So we're going to send you in the mail It's a tube that you can now see on the left. It's kind of a long tube, and you would spit into that tube. And we have videos, we have pictures, we show you how to do this, so it's not hard. And you spit into the tube, and then you put that little cap that you can see kind of hanging upside down, you put that on top of the tube, put it back in the envelope, and if you can see the little arrow, postage is paid, it's already paid, you just put that in there and you mail it back to us. So the answer to that question earlier is that you're going to collect saliva in a tube. And from that saliva, we can obtain DNA for us to be able to ask some questions about genetics. Now, let me kind of walk you through the process. So we covered a lot of different things about what you do as part of this study. And I just want to take a step back, make sure everyone understands what you'd be doing if you participate in the study. First thing you do is you come to our website. You learn about the study, you can kind of look at what we have, and then you participate in that online consent. Depending on the age of the person, you could do it all by reading it if the person is over the age of 14, or if they're between the ages of 7 and 13, they would watch a video. For anyone under the age of 18, though, we're going to need to have the parent or legal guardian also sign a consent saying that they agree that this individual can participate. After you do that, if you have questions, we'll answer them. And then you'll be able to complete some forms online. Remember, those were the forms about demographics. It was about whether or not you've been seen previously by any kind of a healthcare professional. And then remember about prenatal alcohol exposure. Then you take some photos of the individual, and each of these things happen kind of one step at a time. We send you those stickers, then you would take some photos. Remember, 3 different angles, and then you upload those photos. Then the last step is we send you that envelope in the mail in which you would get a tube, and you would spit into that tube and then send it back to us. So those are sort of the 4 main parts of the study. And remember, why are we doing all this? We're doing this to better understand how the genetics or DNA affects the features that we see and don't see among individuals who have fetal alcohol spectrum disorders. And ultimately, the reason we care about genetics, the reason we're trying to understand why individuals have this variability is we're hoping it's going to give us opportunities to think about new treatments or maybe improve our interventions. How can we help individuals who already are having some challenges due to prenatal alcohol exposure? How can we help them so that they can meet their full potential? And that's really what we want to be able to do in this study, is really improve our care of individuals. So I said I would do this for about half an hour. I just want to remind you, there's always faces behind all of this. So there's 3 of us that are kind of the core members of our team: Elizabeth Rowe, Leah Wetherill, and you keep hearing my voice, Tatiana Frood. We would love to tell you more about Dig FASD. We would love to talk to you by phone. We'd be glad to get your emails. Glad to have you, of course, come on our website and learn more. You can see that on the bottom right. We've had it on several slides, but we would love to have you participate in this study, and I would welcome your questions. And so if we can open that up, or if there's questions you can read, I'm glad to start to do that.
Okay, great. Thanks so much, Tatiana. We really appreciate it. And yes, we do have some questions. And again, people are welcome to type their questions at any point. You can continue to type questions, and we'll get to as many as we can. So one person wrote, how can the role of genetics in the study of FASD help adults with FASD?
Oh, that's a great question. So I think probably the first answer is, and let me start by saying something I didn't say that probably should have, is for the most part, we really can't change our DNA. And that's maybe perhaps part of the question is, if we can't change DNA, how can this give us any important insight? Well, one of the things is we don't completely understand why there's variability in features. So if we understood what genes are important, it tells us something about, and I'm going to use a big word, pathways. It tells us what might be going on in the brain, the rest of the body, that helps us identify ways that we can design interventions. So let me take a step back. If you think about medications, the way many medications are developed for disease is we figure out why an individual develops the disease. Once we understand that, we can take a step back and say, how could I change that pathway? How could I change that? And so that gives you a way to intervene. I'm not sure that I'm saying necessarily we're looking for a medication. In many ways, we're looking for, it could be behavioral interventions. It could potentially be other ways that we could understand why there have been the changes that have been observed and think about ways that we can maybe take another path. Can we help somebody cope or manage in a different way if we understand what's not perhaps working as well as it could? I don't know if that, I'd be glad to take a follow-up question if the person wants to ask a follow-up question. So I think about ways that you can intervene not only in children, but I think we're very interested in understanding pathways because I think it is never too late to be considering interventions. And I want to stress that. I think that's very much a commitment of what we're doing in CFAS. We have studies of young individuals and we have studies of adults who have fetal alcohol spectrum disorders. We are committed to looking at the entire age range because we feel we want to be able to intervene and provide opportunities for behavioral changes. We'd love to ultimately, if there were ways that we could develop medications, that would be wonderful. But we want that to be accessible across the lifespan.
All right, great, thank you. And we also have a question about the issue of the minimum age being 7 and there being special tools for, I believe it was 7 to 14-year-old participants. Could you maybe talk about that in consideration of individuals with FASD that might have a younger developmental age and how that might play a role in terms of the consent and comprehension of the study?
A great question. So at first I thought the question was why not enroll individuals who are younger, but I think what you're really asking is if you have an individual between the ages of 7 and 13 and perhaps developmentally they are younger than their behavior might be more similar to a child who is younger. So there's 2 pieces to that. That's part of the reason why we ask questions while the individual is completing these videos. So it also gives us a feel for, they're not tests. I really want to stress, it's not like you need to pass those questions, but it gives us an opportunity to see how well the individual is understanding that information. Importantly, we also engage the parent or legal guardian as well. So one of the reasons we also like to talk to individuals at different phases during the course of the study is it gives us an opportunity to make sure the individual understands what this study is going to involve. And as with any study, there is the opportunity for us, if there is a concern that the individual really doesn't understand perhaps exactly what is being, what's being participated or what they might be doing, there's the opportunity for us to have a question, should this individual be involved in the study? I'm not sure if that's the direction that the individual wanted to go in with that question. If I've taken it a different direction, I'm glad to go in another direction. The other direction for that is, I think long-term, we have partners within the FASD consortium that are actively involved in enrolling individuals who are well under the age of 7. And so we are very eager to explore ways that we can engage younger children in a study like this as well. And so we just wanted to bite off the part that we felt that we could do well, but we're very interested ultimately to be able to involve children who are even younger as well. So I've kind of given 2 parts to that depending on which way the person wanted that question to go. I hope I've answered it one way or the other.
Yes, thank you. And we have another question regarding considering the compounding effects of systematic marginalization people with FASD experience. Is there anything being done to include their data or accommodate them in the study, or will that be included in future studies?
So I want to make sure I understand the question correctly. So is one of the concerns, for example, the internet access, do you think, so that there are some individuals who might not have the ability to participate because it's an online study? That's maybe a question I'm asking you. I don't know if you have a sense if that's the direction of the question.
Yeah, that seems like what they're doing. I'll let you know if I hear a follow-up from the individual who wants that.
So I will say, so one of the challenges we have is, again, we're located in Indiana, and so we do have sites that are part of CFAS that are doing other types of research, not the same research that we're doing, but some of it's related. So if we had an individual who, for example, who was able to call and let us know that they were not able to participate because of limitations, for example, of internet access, one of the things that we could do is, if they were located near one of the other study sites, potentially we could try to involve them in a research study that was there. We could also try to help them identify other research studies that might be outside of the consortium that they might also be able to participate in. There is also the possibility that portions of this study, for example, could be done by paper and pencil. So for example, the forms that we do online, if somebody didn't have internet access, it could be possible that those could be printed and a person could complete those through paper. So there are ways, if we begin to realize that there are significant proportions of individuals that are not able to participate because of the internet aspect to this, I think we are very ready to try to accommodate other ways that they can participate, because I think anyone who's willing to participate in research, we want to make it possible for them to be able to do that, because that's really such a wonderful thing to try to make a difference in this disorder and in this area, that we really want to make sure we make it accessible to everyone.
All right, great, thank you. And another question is, can you speak to the possible role of the male as it relates to FASDs?
It's a great question. So when we think of fetal alcohol spectrum disorders, so fetal alcohol implies, therefore, that it would be alcohol exposure that would occur in utero. So when we think about that, we typically right away begin thinking about mother and child. But I think the person actually has a great question. If you go back to my early slides, half of an individual's DNA comes from the mother. Half of an individual's DNA comes from the father. So any susceptibility, if I may use that word, or any variation that DNA may be playing a role in that makes an individual have more or less findings based on prenatal alcohol exposure could have been inherited from the father. So the father absolutely plays a role in the context of the DNA that's been inherited by the child or the baby that hasn't been born. The other part of that question is, and I'll take this a little farther, is could there be an impact, for example, from a father who might be using alcohol, and could that be affecting, for example, sperm? There's not a lot of research in that area, so it would be probably pretty speculative, at least for me, to comment on that. I am not aware of anything in that area, but that does not mean that there might not be some data in that area. That would be the other way that the father could be contributing to risk. I hope that maybe answers the question. I kind of gave it two parts again to try to make sure I've covered it. Yes, thank you.
And let's see, we got several additional questions as well. So somebody is asking, in their words, that it seems that you're comparing, or the study's comparing, the genetic material to the photos of the face and measuring the facial features. And they're asking, why is that the focus?
That's a great question. So this is actually the first part of the study, okay? So we've actually tackled, I hate to say it, the easiest part of the study first. So being able to take pictures of individuals is actually in some ways the easiest part of the study. The study is right now working with Dr. Sarah Matson, who's located at San Diego State University. And she has done many, I don't want to say many decades, but many years of research looking at if we call them neurobehavioral cognitive changes among individuals who've been exposed to alcohol prenatally. And she does a large number of what we would might call pencil and paper tests to try to document, for example, attentional changes or different kinds of areas in which an individual who might have been prenatally exposed to alcohol might perform differently on those pencil and paper tests to individuals, for example, with ADHD or with similar IQs or cognition. What we're working on right now is actually developing some online instruments so that a person could get this, I don't want to say the same information, but some other information that we learn is important from her studies that we'll be able to do that online. And we hope to have that ready in a year. And I bet many of the people listening will say, aha, that's probably in many ways what people are most interested in, perhaps, is how might variation in DNA be contributing to how someone is performing on kind of pencil and paper tests like that, because in some ways that might have a more immediate impact on how the individual is able to function or how they're able to kind of have their future life prospects. And so we're not quite ready for that. We're working with Dr. Matson right now with that, but that portion of the study won't be ready for about a year. So individuals who participate now are going to work their way all the way through the saliva in about a year from now, they're going to hear from us again, and we're going to ask, will you come back to our website again, and will you now come and participate and complete these new online tests that will allow us to now begin to look at neurobehavior, cognition, things like attention. So that's going to be the next phase of the study, which is going to be about 1 year from now. And I bet you that's what the person might have been thinking. Why aren't you asking questions about that?
Great, thanks so much. And we have a question in asking if you're aware of any information or any research on the role of adult stem cells in terms of adults with FASD.
Oh, that's a great question. So there's a lot of interest around stem cells. So I'm going to say a few words about stem cells, and unfortunately I'm going to say that I'm not aware of anything, but I just want to take it as an educational moment. So many people, when they hear stem cells, think about embryonic stem cells. So those are stem cells that come from fetuses, which can often be a very controversial area. There are many other ways to develop stem cells that can be developed now from other kinds of cells in the body. They can be developed from, for example, skin samples and also from blood samples. So while I am personally not aware of any work right now that suggests that stem cell research would be a way to potentially consider an intervention, it's certainly an area in which there is so much research kind of broadly, not necessarily in the fetal alcohol syndrome field, but broadly, that I wouldn't be surprised if we wouldn't be talking maybe about what we would learn from stem cells and how that might be applied potentially in the future to improve treatments. But I want to stress, to my knowledge, I'm not aware of any research at this time using stem cells.
Okay, great. Thank you very much. And another question is, can you please tell us some more about results of your previous work on the effects of genetics on FASD?
Oh, that's a great question. So we have previously published, and maybe this person is aware of that. So within the CFAS consortium, and it's very much the motivation for this study, we have collected previously largely saliva samples from individuals to be able to look at some genetics. And the work we initially focused on was actually with some of the CFAS collaborators who are working with animal models. So they were working with mice and also with zebrafish. It's a kind of fish that you might actually have in a fish tank. And they, because of the species, were able to do modeling of prenatal alcohol exposure, both in fish and also in mice. And from the work that they did, they had some hints of some genes that they thought might be important. So what we've done in an earlier iteration of this study is we used the saliva samples from individuals who participated in earlier CFAS studies and actually looked at the genes that these other researchers suggested might be important. And for one of those genes, we saw some evidence that suggested perhaps this gene is important. But one of the challenges we saw very quickly is that it was hard to draw conclusions when you only had a couple hundred individuals, humans, participating in this study. And that's actually part of the motivation for the study we're doing now. I opened and told you that we'd really love to enroll more than 1,000 individuals. When you do genetics, it's really about being able to ask questions across much larger numbers. So that's the reason why we've tried to do this online now, because we think we can have many more people participate this way. So again, one of the ways that we'll use the DNA that we obtained from this study is to take a step back, look at some findings that other parts of the CFAS consortium have suggested, and ask, are they also important in humans? Are they also important in individuals who have done the cognitive testing I'm going to told you is coming in the future? And now in the first year, we'll start just to be asking, can any of the facial features that are characteristic of fetal alcohol spectrum disorders, can we tag any of that to potentially DNA changes? And I guess I want to take a step back and ask and kind of remind people, it's not so much the face that we are asking about, but the face reflects the brain. And that's why we ask about smaller head size. That's why the eyes are so important. The face often just reflects whether or not there's been any kind of an impact on the brain. And that's why the face is important. It's not because someone's chin is smaller. It's not because someone's eyes are smaller. That's simply pointing to perhaps something's changed in the brain, and that's an outcome of that. And that's really the reason we use the face. I just want to make sure that people understand that.
Great, thank you. Yeah, that's very helpful. And we have another question from somebody that's interested in getting more information on how the variation of DNA with FASD might affect medication management, and they're asking if using pharmacogenic tools like GeneSight that help guide medication pathways, if that proves helpful in terms of FASD.
That was a great question. So what they're talking about is pharmacogenomics, and that's a great question that someone's just asked. So let me take a step back and exactly kind of take a step back, I guess is the right word. So pharmacogenomics is really important. In all areas, not just in fetal alcohol spectrum disorders. Pharmacogenomics is asking why, when again two different individuals take the same medication, why do they respond to the medication differently? And one of the reasons people can respond differently is because of variation in DNA. So there's, if we can talk outside of this area, people will look at that in terms of the way people metabolize or the way a medication is used up in the body. People will actually metabolize or use up medications at different rates sometimes because of variation they have in DNA. There's also variation in DNA. If some of you are familiar with simvastatin or the statins, which are used often for cholesterol lowering, there are some individuals, because of variation they have in their DNA, who respond poorly to that medication. So the person who's asking this question might already be thinking, if we were to develop medications, or if there were to be some interventions that involve medications, we'd have to also ask, do all individuals respond to that medication in a similar way, or do some individuals respond differently to that medication? And differently could be, there's a medical word for it, adversely. They don't feel well, makes them sick. And so it's very important anytime you do new medications, that's one of the things that needs to be evaluated before a medication is approved, is how often do people have a poor reaction to the medication, what percentage of people do, and can we predict who that will be? And so that would absolutely be the case. And I think that might be what the person's asking about.
Great, thank you. I think we just have another few questions. And one of these is very specific, so I'll ask that. The person asks, we already know that my son has a 1q21.1 microdeletion. Would this make him ineligible for your study?
So actually, no. So we have had individuals participate in this study. So one of the things we have for the study is if you go on, you do the consenting, it asks about previous medical history. So what that individual would do is they would upload that information, that test report. And we actually have a genetic counselor as well on the study. And one of the things we'll ask is, what are the features of an individual who has that kind of a deletion, and will it confuse the results of this study? So in other words, sometimes that particular deletion might give features that are very similar to fetal alcohol spectrum disorders. And if that's the case, it might make it confusing to interpret any data that we have from that individual. So that would be one of the things that we would need to look at. We would do that on a case-by-case basis, and we would talk to the person, and we would be able to explain to them why they can or can't participate. But we would definitely encourage that person to first come forward, and let's have a discussion about that first.
Great, thank you. And we have a follow-up question regarding the face and the face reflecting the brain. And they're asking if you could expand on that in terms of if an individual does not have facial features, what area of the brain might be impacted that causes the behavior challenges.
So that's a great question. So that's exactly the kind of variability that we've been talking a lot about. And it's probably one of the greatest frustrations, I think, among families who have a child or an individual in their family that is believed to have had prenatal alcohol exposure. The face, there's no features that makes an individual come toward a diagnosis. And so as a result, often there's no explanation for behavioral changes. So unfortunately, it doesn't map quite one-to-one. It's not quite as obvious as that. So one of the reasons in fetal alcohol spectrum disorders, we always measure the size of the head. Is the size of your, it's called an OFC, or the size of the head, and you wrap a tape measure around that. That's a really crude way to just kind of ask, how big is the brain? And so it's not a one-to-one. Again, there are some people who have relatively small head sizes and their brains are working very well. It's certainly not an absolute factor, but often individuals who have very extremely small, so we typically say less than, say, the 5th or 10th percentile in terms of their head size, that could be an indication that there might be a smaller brain, and again, that might reflect then in other features. But what you can't do is say, well, the eye is small, therefore it's the hippocampus. The relationship is not as close as that. But for a number of the fetal alcohol CFAS studies, there are sites that are doing brain imaging, also doing a dysmorphology exam where an individual is looking at the face, and are also doing then the pencil and paper tests. And one of the reasons they do that and then pair it with DNA is we're trying to look at what is the relationship. Can we begin to do a little bit better mapping of what's going on in the brain when we look at with MRI, what's going on in terms of those pencil and paper challenges, what's going on in terms of the face. And then we can also ask what's going on in the DNA. So the more information we have like that, the more we can make comparisons. One of the challenges and the reason we're doing our study is to do all of those things for an individual is actually quite costly, the MRI in particular. And so that means a smaller number of people can participate in the study because it's so costly to see them and to be able to do that kind of imaging. So what we're doing in this study is trying to see more people, but we don't try to get brain imaging. So we sort of get more people, but we don't get that look inside what's going on in the brain with an MRI. Hopefully that makes sense.
Yes, thank you very much. And we have a question that came in that is asking if the study makes any differentiation in terms of individuals with prenatal alcohol exposure as opposed to people with an FASD diagnosis, and if there's any differentiation between the different forms of FASD, like the fetal alcohol syndrome or the partial FAS or any of those others, if that affects the determination of genetics in FASD.
That's a great question. So I started with the umbrella, and I said the criteria to be involved in the study was suspected or known prenatal alcohol exposure. So we're actually really interested in enrolling individuals who are across the entire spectrum. We're actually interested in enrolling individuals who have all of the classic features of fetal alcohol syndrome, all the way to individuals who have probably very, very limited features, because that gives us the best way to ask what's the role of DNA in terms of all the variation that we see. And the way we try to, as best we can, put people on a level playing field is if we're able to gather some information about prenatal alcohol exposure, that also helps us layer that on top of it as well. So it's very important for us to see individuals who lie across the entire spectrum. The thing they have in common is that there's been some prenatal alcohol exposure. I think that's what the person was asking.
Thank you, yes. And another question is if you could talk a little bit about popular genetic websites like Ancestry.com and 23andMe, and if that could play in some role in the study, or if those kinds of services could be useful in terms of better understanding the genetics associated with FASD.
It's actually a great question. So I think a year ago, I probably wouldn't have gotten that question. So there are actually a couple different ways. So many people are familiar, Ancestry.com, 23andMe, those are direct-to-consumer testing services. So individuals obtain genetic results, and in some cases, they can actually download their results. And so it's certainly possible that someone could let us know if they're participating in our study that they've also participated in that study. There are ways for people to share information that they've obtained from those services. It's probably ultimately easier for them to actually give a new DNA sample through saliva than it is to actually be able to download the data from those services and try to get that brought into our study. But always glad to know about that. And importantly, there is no, and I just want to stress this, there is no genetic test for fetal alcohol syndrome or fetal alcohol spectrum disorders. When I showed you those chromosomes, what I hope I was conveying is right now there is no test that we can do that looks at DNA that can help us try to, for example, make a diagnosis or anything like that. So we're not at that point. Fetal alcohol spectrum disorders is a diagnosis that, typically evidence of prenatal alcohol exposure or features that are very consistent with what we would expect to see with someone who's been exposed, which is typically the more extreme set of features. So there is no test. There's nothing that 23andMe or Ancestry.com can tell you definitively, yes or no, this is a fetal alcohol spectrum disorder. But what we're hoping in the long term is that paired with evaluations paired with an understanding of whether or not there might have been a prenatal alcohol exposure, we hope that DNA might be able to help us give us a prediction of how could we best intervene, or maybe give us an inkling for ways that, give us ideas about ways to intervene and to provide intervention. So that's where we hope to go, but we're definitely not there yet.
Great, thank you very much. And as we come to about the end, it's almost 3:30. I was going to try to get in one or two more questions. One is, is the study going to be looking at epigenetic changes?
Oh, what a great question. So yes, so epigenetics means changes. They're often described as methylation changes, and they can be changes that are made in the DNA, sometimes due to environmental exposures. So we actually have already done a little bit of work in that area in previous rounds of the CFAS consortium, but definitely something that we're interested in as well, and something when we go back to the question about paternal exposure and involvement in the outcomes, and also maternal, very, very relevant. So it's hard to imagine studying fetal alcohol spectrum disorders without thinking about epigenetics and methylation.
Great, thank you. And I think this is a good closing question. So what are some ways that people can promote the DIG study, help to get more people enrolled? What are the different things that people can do to assist in that way?
So I am delighted to have that question. So I think any opportunities, individuals that are on this call or are listening to this webinar. So if you have support groups that you're involved in, if you're involved in social media, we have flyers that we can send to you if you would like to be able to distribute something in paper. We have some electronic PowerPoint slides that we are glad to provide you if you have the opportunity to use something like that in a talk that you might be giving. Glad to have you put out our phone number, our website, any opportunity you have to let others know about this study, and any way that if you would like want to refer a place that we should be going to try to let more people know about this study, we would welcome that. Feel free to send a link to this this webinar. The key thing for us is getting the word out. We just really would like to have people know about the study so they can choose to participate.
Great. Thanks so much, Tatiana. Before we close, is there anything else that you'd like to say in closing? Any final statements or anything?
Something incredibly important on my slides. I forgot to thank, and I'm embarrassed to say this, I have forgotten to thank on my slides the National Institute, National Institutes of Health, and in particular NIAAA, which supports this study. And without their support, this would not be possible. And I am very grateful to NOFAS, who invited us to come give this presentation. Very grateful to both organizations.